So hello, good morning. Well, I'm from Brazil. I'm working for a few slides about VTT. VTT is a Finnish company. And first, I would like to thank the organizing committee for inviting me to talk here. And well, I liked a lot this city. It's a really nice city. And on Sunday, there was a really fun situation because the taxi driver who picked me up at the airport, he asked me where I am from, and I said Brazil, and he laughed. Okay, but what the hell? And he said, okay, you lost the World Cup. Oh, come on. Don't you have any other issue to talk about? We are hurt. But I'm kidding. He's a nice guy. Okay. Here are the topics that I want to talk about in my presentation. Okay. So, as I mentioned, VTT is a Finnish group, and we have been working in many areas, like you can see in this slide, since biotechnology and also other different kind of fields. Okay, here are the where we are in the world. So mainly in Finland, but we have also a unit in Brazil, in São Paulo, one unit in Berkeley, and other offices along the world. Here are our building in São Paulo. Okay, so in São Paulo, in Brazil, we have a main focus on biorefineries. So, okay, well, I don't work with clinical research. Maybe my presentation will break the sequence, but I hope you enjoy. So our main interest is in biorefineries. So, and now we are making more efforts on biomass hydrolysis, enzymes for that purpose, and also looking for bioprocess for green chemicals and other. Okay, yeah, it's really impossible to talk about biorefinery and in Brazil without mentioning that we have a great biodiversity. And it's really nice because, and now I would say that I'm proud of Brazil because we never saw such big investments like we have there now for science development, especially about bioenergies also. And now, I would say that Brazil figures between the hot spots in the world about bioenergy. Okay, just to introduce, of course, everybody here knows about these data, but well, sometimes we forgot about the evolution of techniques. And, well, the Human Genome Project maybe was a driving force for new technologies on sequencing, and we can see in this chart that, okay, it's an interesting data because we can see before 2000, there was an exponential decrease in cost per base of sequence, and also exponential increase in the quality of base sequenced. And this is also interesting because we can see also regarding the cost per base. And okay, here we have the Sanger era, and we had 454 era. It was really important, and after we have really different new technologies now, Illumina and also PacBio that can sequence a single molecule. Okay, and here we can see the evolution of sequenced genomes since 1997, and also a projection till 2025. So it's really interesting because for biotechnology process, it is very important. This kind of data could provide a really good material for searching new enzymes, new genes, for improving or for creating new bioprocess. So, and now we have a kind of, we are in a new era, and maybe sometimes could be a problem, a kind of problem how to handle big data and a lot of genome being sequenced. So the limiting factor is not anymore, or it should be not anymore, the sequencing techniques, but sometimes bioinformatics is now the limiting factor. So a key point is how to integrate all these kind of techniques. Now we have access, easy access to metabolomics, proteomics, transcriptomics, and some metagenomics, transcriptomics, metaproteomics. And so this is a key point, how to integrate and how to get good information of a lot of data we have produced. So, okay, and now I'll show you the experiments that were part of my doctorate. And so, regarding enzyme discovery in this new era, we have mainly, I will focus in these three techniques that now they have been a lot used. Metagenome, metatranscriptome, and metaproteome. So this is an article that we published, I think this year. And it is about one enzyme that we isolated from metagenome soil. This soil sample was collected in sugarcane mills. So all the work I will show you is related to enzymes that degrade biomass. So this kind of classification is for glycosyl hydrolase. It's a CASI classification, right? So here is the structure of this enzyme we isolated, and this is a nice result because we can see the temperature and the pH curves together, evaluating the activity of this enzyme. And we could see that this enzyme works in a wide range of pH and also temperature. It's really important for biotech process. And also this picture is interesting because this enzyme can hydrolyze oligosaccharides to monosaccharides, disaccharides, and trisaccharides. And it is also desirable for our process. And here we supplemented to the commercial cocktail, we supplemented this enzyme and we got good increase in hydrolysis of biomass. Okay, this is another work where we did secretome of Penicillium echinolatum. This is a good enzyme producer, good cellulase producer. And, okay, the idea is we produced the enzymes. We grow this fungus in 5 kind of substrates. They are all cellulosic substrates, but sometimes the pretreatment is different. Chemical pretreatment, steam explosion, hydrothermal, and in natura biomass. So, sorry, the picture is quite small, but we can see the cellulase are blue, and we had a different expression of cellulase using different kind of substrates. And this table, this heat map, we divided the peptides we found in mass spec by all the substrates we test, we grow the fungus. And mainly it produced cellulase and xylanase. Well, cellulase is the major polysaccharide that we have in nature, in plant materials. And also xylan is maybe the second one, the hemcellulose. And here we characterized the enzyme mixture produced in which substrate and each substrate. In natura bagasse, in natura biomass, the first one was really nice for enzyme production. And it's a good because for industrial process, it could be really important. Another work we did also a characterization of enzyme cocktail produced by Trichoderma raceanum. And we did also proteomics of this secretome. Okay, the major enzyme produced was also cellulase. We can see. I'll not give you a lot of details about these families, but well, cellulase were major produced. And interesting, with this study, we identified that this enzyme mixture was missing mainly 2 enzymes, because for biomass degrading, we need a big set of enzymes. It's not 1 or 2 or 3. It's more than 10, 20, and well. So we identified that it was missing these 2 enzymes, and when we supplemented, we got more than 100% increase using this fungus. And this is a good commercial standard. This is a good standard fungus for cellulase production. This is another standard fungus, and this is also a commercial standard enzyme. So it was really nice because it is a start point for genetic improvement of this strain and or supplementing enzymes. Okay, and the last topic about protein engineering. Well, I really like protein engineering because, well, actually, indeed, we can do most of the time what we want. Of course, not so easy, but if you choose a good strategy, sometimes you can find really good results. Okay. So, and the current paradigms are what strategy? Rational or non-rational approach? Of course, it depends a lot on what you want to do, but sometimes it's better to run both in parallel because, well, you know that for enzyme production, for enzyme mutation, sometimes you think, okay, I'll mutate a single amino acid to have this effect, but when you do, you cannot have this effect. Or so Okay, this last work I'll show you is another. Okay, fine. Okay. This was a really nice work where I built a chimera of 2 enzymes for biomass degrading. So this kind of strategy, multi-domain, multifunctional proteins, can reduce costs with enzyme loads, because the idea is produce 2 domains together in the same polypeptide, and you can save sometimes. This is the idea. But this kind of strategy, sometimes you can have not good result because the protein could be not functional. And of course, in this case, the linker selection is really important, right? So first, this study was, I had a collaboration with Professor Onushik group from Rice University, and they are experts in molecular dynamics. It's not my background, but we collaborated. And okay, basically, this first simulation, we got a point here where we had a good probability for this chimera, because first we simulated computationally this chimera. So this is the CM distance between the 2 enzymes and radius of duration, right? Okay, so based on that, we did experimentally, and we can see here the enzyme, the chimera, and the models in separate. Okay, we did also, after building the enzyme and purifying the enzyme, we did a small angle X-ray scattering. And this curve, it was really, the experimental curve was perfect, had a perfect fit to predicted structure, right? According to the model. So this should be the most probable structure of this chimera. Okay, some biochemical data. About the pH of activity of these enzymes, we didn't have so much change, and also no for temperature. And also about the specific activities in different substrates. It was quite the same, not so much difference. And also about the products of hydrolysis. It was quite the same, not quite the same, it was the same in both cases. And the most important parameter is the enzyme yield. This is the most important thing, because the activity was really similar, but we got more activity with chimera. This is what it means, right? 6 times, in this case, for xylene. Xylene is a substrate. For hydrolyzing xylene, we got 6 times greater, and 20% greater comparing to lignin substrate. Okay. So this work presented a novel way to predict disposal of chimera domains in solution before experimental assays. And this is a potential tool for screening and development of enzyme cocktails for second-generation biofuels or other fields. The expansion of hydrolysis activities in a unique protein could be a road for increasing cost-effective of biomass saccharification. And enzyme production data suggests an advantage on producing the fused protein instead of the wild types once paired. Okay, and just to finalize, these are the most common topics that we can work currently for improving some process and enzymes. So these are the typical challenges that we have. Increase activity, increase thermal stability, and maybe change the activity to another. And that's it. Okay. So I'd like to thank CTB, Unicamp, and thank you very much.
Sure, continue.
All right, thank you very much. I have a quick question. If there are any other questions, please bring the microphone. But before that, thank you for a lovely talk.
Okay.
So very sort of a general question. What is your hit rate for brand new functionality enzymatically? So do you discover sort of a new functionality every week, every month, every, however, either by time or by experiment? What's your...
Well, you mean about the time?
Time, yeah, so what's your experience with finding brand new enzymatic functionality?
Okay.
Is that an everyday occurrence? Yeah.
Well, it's a good question, but it of course depends because, for example, now you can produce some mutants really fast. Especially now, for example, in Brazil, we are doing now some mutants, and we are ordering the genes. We can order, so it's really fast. But sometimes you need more time for computational simulating, and it could be the limiting factor. Sometimes you will spend 1 month or 2 months, and if everything is okay, you can go to the lab and do the experiment. But especially molecular biology now is so easy to practice. I mean, you can order the genes. You can. The most important thing is think about the right way, but not about the time for doing the experiments in the lab is not a limiting factor anymore. It's my opinion.
That's what interests me. So you're making these chimerics, but you don't. That's what interests me because you're making the chimerics, but you're obviously you have to understand the kinetics afterwards, not only just the enzymatic kinetics, but also the degradation kinetics of these enzymes, because you may be changing the structure enough so that the breakdown is so much slower, or instead of having it for an hour or so of breakdown, it could be days, months.
Yeah,
years.
sure.
How do you deal with that?
Yeah, sure. I didn't mention because it was well. There's a lot of results. Maybe later you can check. I can pass you this article, but we did these kinetic studies, and well, actually, if I remember correctly, it was a little bit improvement in kinetics of one of the models, not in both, but in one model. So, but we studied. We can, I can give you the article later, but we did. Sorry, just, and about your question, we also did another chimera, and now we are trying to license this. There are 2 companies interested in, not that one I showed, but another one we patented. And we had a really good result in, we had 5 degrees of increasement in temperature. So it's really important for industrial process also. It was really nice, the other chimera. But I would say each chimera is a different case because it's impossible to predict. Sorry.
No, I just have you said you're collaborating with Rice University.
Yeah.
I come from Houston. I live 5 minutes from Rice University.
Okay.
So who's your collaborator at Rice?
Professor Onushik.
Onshi.
Yeah.
What department? You
I
don't know.
I
Oh,
forgot.
it doesn't matter. Okay. All
Yeah,
right.
because especially I collaborated with one of his students. Okay.
Thank you again.
Thank you very much.